Okay, we are presenting on protein isolation. It is Amir, Trey, Danella, and me, Regine. Our objective is to extract and separate robusco proteins in spinach from other cellular debris, including the membrane, proteins, DNA, RNA, and etc. The introduction. Okay. Small algae to the whale, which is the largest living mammal, are undependent on the sun as the source of all life on this planet. Photosynthesis transforms the sun energy into a more useful form of organisms. The enzyme which initiates the process in the light is called robusco, which is the most abundant globular protein in the world. This protein is important in the scientific world. Therefore, it is important for scientists to be able to isolate the protein for study. Protein purification. Protein purification is a series of processes intended to isolate a single type of protein from a complex mixture. It is also vital for the characterization of the function, structure, and interaction of the protein of interest. The starting material is usually a biological tissue or microbial culture. The various steps in the purification process may free the protein from a matrix that confines it, separates the protein and non-protein parts of the mixture, and finally separate the desired protein from all proteins. This is a diagram for the protein purification in the multi-step procedure. It starts off as the original sample. It then goes to the chromatography, which separates the ions and the proteins. And then you see if the protein is pure or not. And if it isn't, it goes back into the chromatography to separate the ions and the proteins and everything. If it is, you concentrate it and you use it. Isolation. We use 3 different things to isolate the robusco. In the first step, we use ammonium sulfate precipitation. The second, ion exchange chromatography. And the third, SDS-PAGE, also known as sodium dodecyl sulfate polyacrylamide gel electrophoresis. Okay, the first part we started with adding the ammonium sulfate to the solution, which will cause all the proteins to precipitate out of the solution because ammonium sulfate works as a salt. By adding ammonium sulfate, the solubility of the protein in solution is altered. Once the salt is added to the solution, the molecules dissociate and become ions. Polar interaction occurs between the water and proteins, causing the water to attract the salt ions. By adding enough salt, all interactions between the water and protein molecules can be disrupted. In the second part, with the cellulose fast flow column, which is used to perform ion exchange chromatography, in the columns that were used, small beads made of cellulose lined the sides. These beads were covalently bonded to a side chain with a positive charge. Finally, to test how well the protein was purified, several solutions of the protein were used in electrophoresis since the protein was negatively charged. In the hypothesis, we hypothesized that upon successful completion of the first 2 methods of the purification separation of protein, it is expected that P2 high salt and P2 samples will contain the highest amount of robusco, since robusco contains a 55 kD large subunit and a smaller 14 kD subunit. In the methods, our first step was to grab some spinach, blend it with the buffer 1 at medium speed for 3 minutes, and then filtrate it with microcloth. Then we added 1 milliliter of the filtrate and stored it for later use. Then we added ammonium sulfate to the solution to achieve a 37% saturation. The mixture was then moved to the centrifuge for 15 minutes at 4 degrees Celsius, and then another 3.37 grams of ammonium sulfate was then added to the supernatant to achieve 50% saturation. Then solution was then stirred for 15 minutes and then centrifuged again for 15 minutes. After a centrifuge, 5 milliliters of the supernatant was stored in a concealed tube and labeled as S50, and then another 4 milliliters of distilled water was added to the pellet, stored in a concealed tube, and then labeled as P50. The pellet of the centrifuge solution was resuspended along with 4 milliliters of DI water and then transferred into a dialysis bag labeled P37. Then, in the second part of the ion exchange column chromatography, there were 3 milliliters of diluted P1 and undiluted P2 transferred into the separate columns and washed for 10 milliliters of buffer A. Then each column was loaded with 10 milliliters of low, medium, and high salt buffers. The low, medium, and high salt concentrations contain buffer A along with 50 mm, 200 mm, and 500 mm of NaCl respectively. Next, 200 milliliters collected from both P1 and P2 solutions were transferred into 4 separate Eppendorf tubes for each salt concentration mixture. Each tube was scanned with the spectrophotometer at 200 nanometers to detect proteins. At the end, the column was washed for 10 milliliters of Resign cleaning buffer. This is a graph or diagram to show what happens in the chromatography. This is where it's first loaded, and then as it goes down into the column, the proteins, they bind and exchange. And then when it washes out, you elute it either by retention time or by competition. The SDS page. Okay, this is when we took the samples containing all the high levels of the protein were collected for use in SDS page. From each sample, 60 microliters were transferred into a new microfuge tube. Next, 30 microliters of 3 pounds of sample buffer were added to each tube. To the nature of the protein, the resultant 90 microliters mixtures were boiled for 4 minutes. Then 30 microliters of each boiled sample were added to the following liquids: filtrate S1, P1, P1 medium salt, P1 high salt, S2, P2, P2 medium, and P2 high salt. Finally, electrophoresis were conducted at 110 volts for a duration of 50 minutes with the further analysis of the gel under UV light. Okay, in the results, we used the ammonium sulfate precipitation to perform an S1 sample and filtrate. The P1 and P2 were used in ion exchange column chromatography. SDS page to perform on filtrate, S1, P1, P1 medium, P1 high, P2, P2 medium, P2 high, and S2. Absorb inspection was performed to filtrate on filtrate, S1, S2, P1, P1 medium, P1 high, P2, P2 medium, P2 high. Okay, these are our results. The low salt, our highest was at 0.003. It's a low salt P1, and the low salt P2 was at 0.028. The medium salt P1, its highest was at 0.005. The medium salt P2, the highest was at 0.068. The high salt P1, its highest was at 0.044. The high salt P2 was at 0.439. This is our SDS page. Okay, this shows where at the 55 kD and the 14 kD right here and around right here, and it shows where the Robusco is shown. So at 14 the Robusco is shown, and at 55 the Robusco is shown, and you can see that here, right here, and right here. Okay, in this graph, in the absorbance spectral analysis of Robusco, you can see that its high peaks at P2 high, and that's around 300, well, maybe 280, 300, and the P2, and they're both around the same. That's what the peaks are at. Okay, the discussion. The purpose of this lab was to isolate Robusco, which is the most abundant globular protein in the world and most commonly found enzyme in plants, such as spinach leaves, among a variety of methods to isolate Robusco. The methods utilized in this particular isolation included the ammonium sulfate precipitation, the ion exchange, column chromatography, and SDS page. We hypothesized upon successful completion of the experiment that P2 high salt and P2 samples would contain the highest amounts of Robusco, since Robusco contains a 55 kD large subunit and a smaller 14 kD subunit. Since Robusco contains a 55 kD large subunit and a smaller 14 kD subunit, it is expected that fragments of Robusco appear in the samples at these particular markers. According to Figure 1, samples F, P1 and P2, medium and high, expressed bands approximately 14 kD in the gel. Sample P2 had a band between 14 kD and 30 kD, which indicates it contained dimer of small subunit. The samples that appeared to contain the largest levels of Rubesco are the P2 and P2 high saw as expected. This was confirmed with absorbance testing and by the presence of the obvious bands in the gel. Therefore, the experiments were successful in identifying Rubesco in the spinach leaf samples. The absorbance spectrum results, as shown in graph 1, also support the hypothesis. Graph 1 showed P2 and P2 high with a strong peak between 280 nanometers and 300 nanometers, which is a signature absorbance spectrum of Rubesco. According to graph 1, samples P1 S2 shown a weak peak within that range, which may be attributed to low concentration of proteins in these samples. The STS page data and the ion exchange data both support the absorbance spectrum data, which measured the containment of high protein concentration in both P2 and P2 high samples. The conclusion. As we expected, both P2 and P2 high salt samples appeared to have the largest levels of Rubesco, according to Figure 1, STS page and Graph 1 absorbance spectrum analysis. Purifying Rubesco provides a significant tool to understand how the active site of the enzyme has functioned and evolved. Accordingly, such experiments have the potential to greatly impact in enzymological studies. This is our references.